We'll start by introducing Synapse Life Science Connect Sweden. So Synapse is a student-driven nonprofit organization in the Nordics. So currently it's it's a big organization in Denmark, based in all major Danish universities. And Synapse is quite new in Sweden. So at Synapse Sweden, we're putting a lot of effort at growing this network throughout the whole Sweden. So Synapse creates events, workshops, and networking opportunities for students and recent graduates with an interest in pursuing careers in the life science environment. And the vision of Synapse is to expose students to opportunities and to support them in making informed career decisions by bringing life science peers together. How Synapse does that is by inspiring, developing, and connecting students. Synapse inspires participants through knowledge lectures like this one, and then career fairs and meet and greet events. Furthermore, Synapse develops participants through workshops, competitions, and mentoring opportunities. And lastly, and very importantly, Synapse connects students by providing a platform for people to meet over mutual interests. And very importantly, we connect the future of life science. So all of this wouldn't be possible without our great volunteers. So Synapse Sweden currently consists of 11 volunteers and who are based in Lund. And also we very recently launched a Stockholm hub, which currently consists of 4 students. So wait for further news and from Stockholm. And if you're based in Stockholm or you know someone from Stockholm, keep an eye on our social media. And where to find us on social media. So we are active on Facebook, LinkedIn, and Instagram. And just search for Synapse Sweden in the search field. And you will definitely find us. And it's if you want to stay up to date, definitely follow us. So Rafsan will give us now introduction to Hub AI. So the control is yours now.
Thank you, Enrica. Hello, everyone. My name is Rafsan. I'm the co-head of liaison at Hub AI. So what is Hub AI? It's a student-led network which has the primary goal of bringing people together from all disciplines. So AI and machine learning, these topics are usually quite exclusive to computer scientists and researchers. However, at Hub AI, we try to demystify AI and bring people together to educate, connect, and build, because AI is a powerful tool that we can use for to find solutions for everyday problems. Yes, so here we have our lovely team, and you can also join us because we have an open position for vice president. You can of course reach out to us after the talk to learn more about this position. The events we had before, we organize different types of talks, discussions, pizza parties, everything else. So both online and offline, because we were actually established during the time of COVID. And so a lot of our events have been online, but recently we have been starting to have more and more in-person events. Please connect with us, and you can become a member. And of course, the membership is free. Yeah, welcome to reach out to us at Hub AI over social media. Thank you. Back to you, Enrica.
Thank you. Yeah, thank you, Rafsan, for a great introduction. And now, before we start, we thought that it would be great for our speakers to know more about the background of our audience. So I will launch a poll which about your level of education. So I think you should see your field and okay. A lot of answers are popping in, and okay. Yeah, thank you, everyone. So we have majority is master students, and then almost equal proportion of bachelor's and PhD students. Thank you. And then we are super interested in your educational background. Here, so yeah, majority is life sciences, then STEM, I guess it will be engineering and computer science, mathematics, and then yeah, some other students from other fields. Thank you everyone for voting, and we will continue with our program. Yeah, so this evening we have 3 speakers who are professionals in the field of artificial intelligence and life science. They will give you an introduction to this field, will inform you about their career and their research, and will present you about the current opportunities. So firstly, we will hear from Marzieh, who is a data scientist at AI Sweden. So Marzieh graduated from Umeå University with a master's degree in computational science and engineering, and now is a part of AI for AI, a graduate program for future leaders in artificial intelligence at AI Sweden. Currently, she works as a data scientist on predictive modeling using deep graph neural network for AstraZeneca, and she enjoys using her skills to contribute to the exciting AI-related project every day. So hi, Marzieh, and thank you so much for joining us.
Hi, everyone.
Hey, yeah, excited to hear from you.
Okay.
And I will stop my screen share so that you would be able to share yours.
Yes, absolutely. And hi, everyone, and a happy International Women's Day, first of all. And let's start the presentation. Perfect. Perfect. So do you have my screen, right?
Yes,
okay.
We do.
Okay, perfect. So really happy about this event. And this presentation I want to show you is all about the introduction or basic about the AI and the impact of AI in life science, and then look at the AI student and opportunity for students, new graduated or even PhD students to engage more in this field. So, you know, there is a lot of buzz around AI, but what is the AI exactly? I really want to look back to the earliest and purest definition of AI because it's avoid to any confusion. And so we need a little bit of history, but it's not that much. The idea of thinking machine comes from Alan Turing, right? But he has stopped his process because of the computation problem and problem of storing information. But in 1956, in the historical Dartmouth Conference, the Alan Turing theory was accepted and proved. And for the first time, McCarthy coined the term of artificial intelligence. So here what McCarthy proposed. Every aspect of learning or any other feature of intelligence can in principle be so precisely described that a machine can be made to simulate it. So we need a little bit translation here. It means a computer will demonstrate a form of intelligence when it learns how to improve itself at solving a kind of problems that only humans do with our natural intelligence. But it's not enough for me, this definition. I want to look at the factors of intelligence and the definition of the intelligence as well. And the best one is Jack Copeland. He describes some of the most important factors of intelligence. One, general learning, then reasoning. Reasoning like a human to draw a conclusion appropriate to the situation. Problem solving, simply give such and such data to find X. Perception, analyzing the environment, the feature and relation between them. Nowadays, we can see it in the self-driving cars. And finally, language understanding. So we understand AI, an intelligence term. It is good to know that there are different types of AI in terms of approach. Maybe you heard about the weak AI or strong AI, but I want to focus on the middle ground between these two. This one is the stage that we are now. We are using systems that inspire by human reasoning, but does not have to stick to it. What I mean is read a lot of information, finding a pattern and build up evidence such that I am X% confident that is the right solution to your questions based on the information that you fed me. This is the stage that AI currently, we are having AI today. And what has been changed? Because we see the progress from 1950s, 1980s, the deep learning comes, 2000, we see the AI field improvement. Nothing changed about the AI fundamental, but AI function and ability has progressed thanks to computer power, advanced algorithm, and increase of data. So the AI today simply is neural networks plus computational powers. It means combining large amount of data with fast advanced algorithm that the system find a pattern and feature in the data and learn from those elements. And why we need it and why we need to use the AI in nowadays is that we are facing many challenges that we are not solving yet globally, like climate crisis. So we need a new way of thinking, new tools and resources. AI is not the end answer, but is one of the resources that human can use it. And impact of AI in life science. Even though we cannot predict when certain AI product or technology will become available for us, but AI is become a very big part of our lives and also impact in life science. Currently, the AI and ML in this life science mostly used for mining information from the data, finding patterns in the data, building predictive models, accelerating simulation, performing causality analysis, and performing generative modeling. Like 3 examples that I show right now, you can see it. Is like using AI models for microscopy image segmentation, doing DNA or RNA, motif mining, predictive protein, or generating drug molecular structures. And there are more examples if you want to bring it up. So we know about the all benefits, but they are the concerns as well. I'm really fascinated about the ethical issue around the AI. So if you are also like me, really interested, please reach out to Ethics Lab at AI Sweden for more information. And also there is another concern that comes from the fear of the humans, because human always on the top of the food chain, right? Not because of we have a sharp teeth or we have a big size, because we have intelligence. And the question like, what will happen if you are no longer the most intelligent beings? Then fear comes up. So these 3 topics in the TED talks, I really like it. And I want to wrap up these 3 talks and say it. The danger of artificial intelligence is not that it's going to rebel against us, but this is going to do exactly what we ask to do. So it means that if we want to have a healthy system, it means keeping the people in that loop and partner with AI and overcome to our laziness and do the AI everything that we want to do. We want to be a partner and have a healthy system and transparent AI that you can check on top of that. And the AI Sweden and the opportunity for you guys. AI Sweden is funded by the Swedish government via Vinnova and by the public and private sector. And the mission is increase the use of AI for the benefits of everyone living in Sweden or across the world. We have 4 strategic cornerstones laid the foundation how we want to accelerate the use of AI. And each of these strategic cornerstones include a number of initiatives that AI Sweden is developing and working on together with the partners. For example, partner network, we have over 100 from the public, private, startups, and academia. And in this network program, we believe collaboration is the key. So we provide platform with diverse set of perspective, knowledge, and experience to identify common needs and challenges, both from the organization and individual perspective. And it's a productive way to drive joint efforts and share the solution. And then shareable knowledge and resources. It's the best way to advance AI development. We have applied AI as the name. It's on working on the operation of the AI, making sure that the AI method can be used in a scale, in a critical system, in real world scenario. And also they are working on coordinated 6 funding for collaborative projects. And also we have a hackathon and challenges for a student and a graduate that they can want and join us. Please reach out to AI Sweden and learn about the previous challenges, as one of them from AstraZeneca and life science, and the other one was about Zensiag autonomous cars. And Data Factory, you can bring their own challenges and take part in the exciting project or experiment in the testbed environment. And AI transformation focus on the organization and help them to fully embrace and integrate it to the AI to create real benefits, because it's not only about the AI and technology and the data, it's also about the organization, the culture, the competence and ecosystems. And then addressing the talents. And many companies, public, private, or startups, always delayed implementation of AI application due to lack of AI talents. So that's why AI Sweden make an effort in August 2021 to build different talent programs to help that issue. We have several talent programs. I will just choose some of them, but please reach out to the website and learn about the upcoming talent program or current ones. For example, Junior AI Change Agent program is a collaboration between AI Sweden and Hyper Island. And this program consists of 2 periods over 6 months, 3 months for learning AI Change Agent training and 3 months internship at the Selector AI Sweden partner. I for AI program or International Graduate Program is a program that's supported by a Woman in AI Sweden and Human Tech Foundation. And this program is 18 months and the candidate spends 6 months, and each organization and minimum partner is 3, and you can taste different AI related projects. And also you have 1 day at AI Sweden to become a part of the Swedish AI community. In this program that I am currently now involved, I am working with AstraZeneca, Sahlgrenska University Hospital, and Zensiag. AI in Mind is the program that's a collaboration between the Compitech and AI Sweden for PhD students, for master degrees to be part of the AI projects. And if you want to learn more, please go to Compitech Talent Program website. And this program in 18 to 24 months. And we have a long talent program for high schools and AW academic for AI for people that doesn't have any background and wants to build a AI career or portfolio. So we help them in 4 months program. Thank you very much. And please reach out to our website, the YouTube channel, and LinkedIn. And if you have any question, please contact me, and I will help you to connect with someone that help you in the process to find a better program for you guys. Thank you very much.
Thank you, Marzieh, a lot for such an interesting presentation. It was really interesting to hear an introduction about artificial intelligence and examples how it can be used, as a big proportion of our audience is from life sciences, meaning they study biomedicine, molecular biology. And we don't learn actually that much as of right now about computer science, bioinformatics, or artificial intelligence. So super interesting. So some questions that we have for you for the discussion. So at Synapse, we're super interested about the career journey, and maybe you could share what challenges did you face along your way from your like student or general career until you ended up at this super interesting I4AI graduate program?
Sure. If I want to share my story, it will be so long, so I will say it in short and give you more information details for students. So I studied applied mathematics first, and then I tried arts for 2 years, and then I decided to start the computer science in University. Actually, this art helped me to go for the AI because I want to create something and make something out of my programming. So this was a lead, and I am really proud of my art side as well. And then I started to have a practical information beside my theoretical information that came from University. So I go for Uminova Innovation, maybe at University. So I start my startups. And after I started my startups and I realized how I like about to be in a industrial, so I try to work with the Siemens Energy company. And after that, I work with the startup program Homeowner AB in Gothenburg, and then ended up to the AI Sweden and be part of I4AI program. And currently I am a data scientist at AstraZeneca, and I really enjoy every day that I work in this company. You have a lot to engage in life science if you're really interested in this topic.
Thank you. What do you particularly like about your everyday job at AstraZeneca, if you could share with us?
Is that when you're working, I just compared to show you really difference. When I work with the Siemens Energy company, it has a problem related to industrial or the gas turbine. So it doesn't matter how much accuracy you will get. It's a little bit higher or a little bit less, it doesn't matter. But in the life science, you are in the process of drug discovery, drug development, and you are making a change in life of people. So you need to have the AI solution base that has a really great result. So it's a tricky part because AI is not always a good solution for any problems, but help you to read a lot. And that's I'm just missing in my life. And when I started a job at AstraZeneca, helped me to read a lot articles and they perform new way of modeling AI algorithms through the life science. And it's really unique and new topics using AI in the life science. For example, my job is focusing on the crystal structure, and I use the deep graph neural networks. And for me, it was a challenge to find papers related to these topics. But it was really interesting because the team is really educated and high potential, so you can use them, learn every day from them. And I don't know how to say it. It's really interesting. Not all, your day every day is not the same when you are working as a project, because you need to bring in new ideas, new way of solving the problems, and it's like a testing and proving. So it's a research way using AI in AstraZeneca.
Interesting. Yeah, thank you for sharing.
You're welcome. I hope it brings open doors for students, new graduated, or even for PhD students to join to our programs, talent programs, and build their career, because it's really worth it.
Yeah, I fully agree with you. And then another question is about your graduate program. So now you're doing a 6 months rotation at AstraZeneca, but do you know where your other rotations are going to be? Do you decide yourself, or is it the organization AI Sweden that offers you some positions?
That's a really good question, because the first round of this I4AI program was in April 2021, and the plan is already decided to, we have 3 partners in that I4AI program. One of them is AstraZeneca, and then Sahlgrenska University Hospital and Zensiact. So we have a 6 months in AstraZeneca, and it's coming to end really soon. And I will start in 10 of April working with Sahlgrenska University Hospital. And then on August, we will start working with Zensiac automation car. Yeah.
Okay, interesting. And as you will work in the hospital, I guess the project will be about collecting some specific patient data. What kind of ethical concerns do you face in your everyday life as a researcher in artificial intelligence? If you could share with us.
Yes, privacy, and privacy. It's a huge problem, but nowadays it's not a really problem. For example, in an automation car, I want to say it because it's not really related only in the life science. The cars, we need finding images, and it's not possible because the privacy is also included in automation car. So we use the bird instead for finding that a lack of information. So nowadays, if you have a privacy in the data, in the life science or in the patient, you can use another source of information like pet information, and you can just have and build that model and then just overcome these privacy situations. But if I want to say, it's all about the privacy, and it's really hard to not follow up that because it's really important part as well. So we need a regular, informations or rules for that. So AI can be able to use in that healthcare or life science problems. But nowadays we can see a lot of other alternative to solve this privacy issue. But yes, I think more privacy, I just handle it. And also because you're working with the life science problems, you need to be careful about your results because it affects the lives. So that one is also matter. How much you work on it's you, because as in my presentation, AI is not that much smart because it's done what you actually ask for it. So it's your responsibility about that as well.
Yeah, I fully agree with that. And how the AI really organization really goes into these questions about all the ethics. So we've had super interesting discussions with them, and it's been super fun to hear from you, Marzieh. And we have some questions from the audience, and in the last 5 minutes we will go through them. So one question that we had is, according to you, what is the biggest challenge for companies to leverage or implement artificial intelligence, data and algorithms, business models, or regulations?
Okay, that's one question, but a lot of questions inside it. But that's a good one. What is the biggest challenges for companies to implement AI? One thing is that when you're having a project related to AI, you need to have a chart to understand this project that you are using the AI is the best way for answering this problem, because AI is not always the best way or the only way to address the problem and solve that. So it's really good to find the objectives and how much the cost reduction or what it actually solve by having this project related to the AI. For example, one project maybe is not worth it that much to using the AI for solving that, or maybe we don't have the information yet in AI, so why we need to invest using the AI for that project, because one of the biggest challenges is AI is mysterious yet, and nobody believes that AI ability to solve everything, and it's really hard for the top peoples in the business to invest on that, because you cannot show them the past and without any shadow to them. So it's really hard to make investment all the projects to solve by the AI, but it's shows the improvement on accuracy and better decision making. So it could be a partner with the people that are scientific or researcher to help them in the process of better decision making. Not yet is yet challenges for companies to accept AI for every problems to solve by them. Yeah, but it's a progress, so we need to work on that.
Yeah, definitely. Yeah. Yeah. Thank you for an extensive answer and for educating us on this topic. And then final question, and this is an interesting one. How was your experience when you shifted from a computer science from computer science into life science? And was it difficult to work in the medical sector without no prior background about it?
It's my opinion. I don't know what others maybe think, but AI or artificial intelligence, when you want to use it as a data scientist, you are working with data and you must be able to have this knowledge to use different algorithms based on your objective you want to solve it. And having a background of like chemistry or a life science, it's a plus and a huge is a plus for you if you have it, if you want to use the AI for solving a problem, but it's not for me a priority because it's about knowing how to use the algorithms based on the problem or objective you want to use it and solve it, right? So for me, it was not a challenge. Actually, I learned from it. I don't have any background in chemistry, but right now I know about the crystal information. I know about the CIF or crystal information format. I know about the unit cell. I know about the molecular or the dynamic or DFT, density functional theory, and calculating of energy formation from the crystals. So, but if you don't have the basic information and the good level of AI algorithms, you are not able to solve any problems if you have a really good information in life science or chemistry. So I think having a background of something is a plus, but having the knowledge in computer, having a knowledge in the algorithm, machine learning and deep learning, and always updated by learning and educate yourself with the new papers, that one is for me is more important.
Yeah, thank you. And yeah, we think that these days is what is really important is how quickly you can learn and how curious you are, right? And the future of medicine is basically merging many fields into one, many different experts from life science, medicine, computer science. So this is the future of innovation, I guess. Yeah. And so there's unfortunately no more time for any further questions, but thank you very much for
You're welcome.
For such informative presentation and for really great answers. Thank you,
perfect. Just to be wrap up, I'm really respect everybody that wants to build a career in this field. So if you have any question or any help, please reach me in my LinkedIn or my email, and I will help you with that process as well. Thank you very much.
Thank you. Okay. So, sorry. Our next speaker is Esben. So Esben completed his PhD in computational chemistry at Copenhagen University back in 2008, and he has since worked both in academia as a postdoc, in industry as an IT specialist, as well as a self-employed pharma IT consultant. And in 2017, his independent research resulted in several contributions to the chemistry of deep learning renaissance. He joined AstraZeneca back in 2018, where he currently works with development of de novo design algorithms and deep learning assisted retrosynthetic planning. And also, super interestingly, he is the lead blogger at cheminformania.com. So hi Esben, and you're very happy to have you here.
Yeah, thank you.
Thank you.
So I will talk a little bit about life science AI at AstraZeneca, at least a little part of it where I work. So I have a subtitle, Creative Algorithm Designs the Drugs of Tomorrow. But Enrica already introduced me, and it sounded all nice and fancy and such, but I can understand a lot of the audience is actually looking into starting their career, and I can say it didn't always look so straightforward or so progressing in the right way when I actually did things. I finished my original master's was in pharmaceutical sciences, so nothing with computers or algorithms or other things at all. I had an interest for computers and how they worked. But then I did my PhD in computational chemistry. So I worked a lot with molecular models and automated docking into receptor structures and a lot of different computational chemistry algorithms. And then I did the big mistake of finishing my PhD right into the financial crisis in 2008, and all jobs just disappeared overnight almost. But I was lucky after a couple of months or half a year, I can't exactly remember how long, to land a job in an IT department, where I worked as an IT application specialist for 2 years. But after 2 years, I had enough of IT, and I needed, there was not enough science for me, there was not enough research. So I simply had to find something else to do. So I started out without a job, trying to start a postdoc career in protein NMR. So also there was a bit of things. I managed to succeed getting funding for 1 year for a 3-year project and not getting funding for the rest of the project. So it was a complete disaster. But I maybe learned a lot from it when I look back on how to try and raise money. So after that, I was again having some internships. I think it's a nice way of saying that you're working for free on unemployment support. And then I managed also to secure a new postdoc for 1 year, but because there was some leftover funding from another postdoc, and there I learned a lot about chemometrics and PCA and PLS modeling. And also MATLAB programming. I didn't know MATLAB before I started, so I learned MATLAB programming there. But then in the end, I had a part-time consultancy for IT I had been running. So I started out to go full-time there because a former collaborator called and asked me, I need you part-time. I can secure you 50 hours a month as a consultant. And I thought, okay, this will provide the basis for me. So I go full time with my own company, and I bring it up from nothing into a successful company with international clients. And I also get time to do research into various deep learning algorithms by the side, and that leads into the job I have had for the last 3 years here at AstraZeneca as a principal scientist. And now I'm also will actually join a startup in UK and US. And work there starting next month. So yeah, the career can come in all different ways, and it is not always as straight as it looks on paper. That was maybe my conclusion here. So a little bit about AstraZeneca. We have it's an international company. We have a couple of different research sites. A total of $6 billion is invested in research and development across 5 countries. And I'm here from Sweden in the Gothenburg site, which is one of the larger sites in research and development. One thing we are working with here at the Gothenburg site is drug discovery and development. And this is a quite long process. And where I work with drug discovery, it's just the beginning. And it's maybe actually the cheap end. It gets more and more expensive the more towards the end you come, and you have to do all the clinical trials. So it's a little bit humbling actually for me to look at this slide, because I sit maybe and think, oh, this is so fancy, we do all these things, right? But there is a whole value chain actually afterwards that also needs to be in place. And there's a lot of research and development going on in all aspects of the drug discovery and development process until you can actually go from the idea of how we could treat disease until you actually benefit patients in the end. So one of the things we have been interested in, and now we are talking a lot about AI and computer algorithms and anything, it's going to revolutionize the drug discovery process. And actually, if we look back, we have been using computer models for molecules for many, many decades, since the first ones were developed in the 50s, where we take molecules and then we try to predict just by drawing the structure, how will this molecule behave? What kind of properties will it have? Will it be soluble in water? Will it be not so soluble in water? Will there be any toxicity issues? What kind of drug activity will it have on this particular target and other molecular properties? So data and the use of data and computer algorithms have actually been, I don't know, have been used a lot in drug discovery, because we are really interested in optimizing the very, very expensive procedure. And if we can make early failures that makes us not develop on compounds that turn out to not work or have toxicity issues, if we can make the mistakes of predictions very early with computer models, we can save an enormous amount of money and animals and other things in the later stages of the process. But what is actually happening, I would say, here in the last couple of years, is that this process now has begun to be turned around. We are not only using neural networks, or you can say pattern recognition AI, to talk about the properties of the molecules. We're actually taking the properties and getting the machine on the algorithms to design the molecules for us. So we kind of do the inverse of what we have done the last couple of decades. And here, deep learning has really spurred a renewed interest in these. Before that, there was an interest, but you were using genetic algorithms and other combination approaches to this. But really, with deep learning, we have gotten a tool now that works so much better. So what is it we are doing? So we actually took this also quite old molecular format called SMILES. It stands for Simplified Molecular Input Line Entry System. And that was a simple way of drawing and typing into a database a molecular structure, or saving it in an Excel sheet. You can easily email it to a colleague, other things, and it contains all the information you need about the molecular graph. It uses brackets to denote branching in the structure, and if you need to have long-range bonds for ring closures, other things, you can use numbers to denote these ring closures. So yeah, a couple of decades old SMILES, we take this format and say, well, this format, it's a sequence. Let's try and use some of the deep learning architectures for sequences. And one of the things that I started combining it with was long short-term memory cells, which is an amplified form of recurrent neural networks. That means you give it a computation and may do a prediction, and then it remembers an internal state. It can remember sort of what did it do that will influence the next computation. So even if you have the same input again, it may actually give it a different result, because it will also be influenced with the previous prediction that it made. And we can train this on SMILE strings, and this is what we tried out. We have a start character and say, predict the first character or the first token if you use different tokenization schemes. And then if you have the start token and the first token, predict the second token, and so forth. And you can efficiently train this on the LSTM cell. And then after that, you will have a neural network that can predict the next characters for a given SMILE sequence it has just seen up to the point of the prediction. And this is interesting because suddenly when we get the output from giving it the input, it's not a character prediction, it's a probability distribution over different characters, meaning we can sample from this probability distribution. We can say, okay, yeah, carbon atom, high chance, oxygen atom, not so high, and so forth. And we can choose from this. And then we can say, yes, the choice we sampled was right. What would you then predict from the sequence we have sampled so far? We can look at how the process works here. We see to the left the probability distributions as they develop throughout the computation, and then how we build up the molecule at the end. And we see this very typical pattern, actually, that some areas we have a wide range of characters available, but there are other areas, as example, when it builds this 3-fluoromethyl group, where it's actually much more certain about what it's doing. I'm building a 3-fluoromethyl group here, and it's a common pattern in the molecules we have trained it on. And that means we can take a lot of molecular structures and we can have a training set, and we can teach our deep learning network to generate new structures. The surprise is there actually that it not only learns the syntax of the smiles to a very, very good degree. I think it learns it much faster than a bachelor student would learn it. But it also learns the distribution of the data set. So if you train it on small molecules, it will output small molecules. If you use large molecules, it will output large molecules. If they're very lipophilic, it will output lipophilic molecules. If they're hydrophilic, it will do that. And it's actually very difficult to distinguish the properties of the molecule that come up from your input molecule. So that's one way to define the chemical space that we want to generate here. Suddenly we are doing things in a new way, and we can handle much bigger molecular output spaces than we're used to. Some of the traditional ways of doing this kind of virtual enumeration is you take a lot of fragments of different parts, and you can put them together in this way. And if you have enough different fragments, you end up with very huge numbers of combinations to combinatorics. But then you need to store these things you do explicitly on a database. And if you take, one of the biggest data sets you could possibly handle with a supercomputer, you would probably end up with something like 1,000 billion molecules, 10 to the power of 12. So that's a really huge data set you would consider in an explicit instance, right? But that is actually quite a very small number when we compare to what we think is the drug-like chemical space, we call it. If we put up some rules, what is a drug-like molecule, a small molecule for putting in a tablet, it must fulfill these rules. It can combine of these atom types. It must have these and these properties. You end up, depending on the rules, between 10 to the power 30 to 10 to the power 60. And to put that in perspective, I tried to recalculate these numbers into hydrogen. And the database, the traditional approach, we end up with 0.00 nanograms, 70 nanograms of hydrogen. And I put a magnifying glass, but you would not be able to see that at all. It is nothing, right? But if you take 10 to the power 60, you would need more hydrogen atoms than we have in the sun. And that's huge. I have a little illustration here, this planet size Earth to the sun, right? So, but we can still store the rules about how to create these amount of different molecules in a fairly small deep learning model. So previously, a lot of people have said, oh, drug discovery, it's, the needle in a haystack problem. You want to find exactly the right molecule with exactly the right things among a lot of different molecules. And now we have used deep learning to transform this needle in the haystack problem into a hydrogen atom in the sun problem. So we cannot get our hands down. Yes, now we really have a huge space to search in. So what we are doing is actually, because we cannot possibly sample all molecules and evaluate them one by one, what we are doing is we are combining this with other algorithms. And in this instance, we have had very great success with reinforcement learning. So we have different scoring functions. They can be traditional machine learning models or chemical models or various other docking models. You name it, as long as they're not too slow. We can use them, and we put this into a kind of scoring function, a desirability function. This is the properties we want of our molecule. And then we have our generative model. We are trained using recurrent neural networks. And then we use reinforcement learning. We ask it to create some molecules. We score them. We feed that back with reinforcement learning. It creates some new molecules that's a little bit better. And so, and a couple of 1,000 cycles later, we start to generate molecules that have the exact properties that we're interested in. And we can easily generate, say, a data set of 50,000 interesting molecules for a given drug discovery project that can then enter the system. Computational chemists together with medicinal chemists sit and look at them, get inspired by them, group them into different things. And this is a way to use AI to generate the drugs of tomorrow. The system itself, we have actually open source from AstraZeneca. So if you're interested, you can go to Molecular AI, our GitHub repository, and download reInvent and start to generate the molecules that you are interested in if you have a project where you need to design molecules. So one thing I was thinking about now, given the audience, is what kind of opportunities are there for people who have just maybe taken their masters to come in and start to work there. And one of the things I've seen in the last 3 years is that we have this graduate program at AstraZeneca as a kind of entry-level program where you could come with a typically master's degree, and you get then 2 years with 3 different rotations of 6 months in 3 different groups or departments, where you get to work on some of the projects that we are interested in. And it's a kind of attractive resources for the traditional for a research group like ours, because it's quite easy for us to write a project proposal for the graduate program for a project there, and then if it's interesting enough, we can be lucky to have a graduate scientist that can work on it together with us. For this period. So personally, I've been involved in supervising 3 from the data science and AI program. And I think all 3 projects, they were successful. We ended up publishing from all 3 projects, and actually also Panagiotis, we ended up publishing in Nature Machine Intelligence from his project with us. So that's definitely a thing. There have just been a call for this graduate program, but there are regular calls also next year. So there's an opportunity there to look at. And another opportunity that is of course a PhD project. And one of the things that I've seen where there seem to be regular calls is this WASP, which is the Wallenberg AI Foundation for autonomous systems and software. They have this regular calls for postdocs and also PhDs, where we together with a potential PhD student send in project proposals for this, and we can then get some grant money. It needs to be a collaboration between an academic institution and an industrial placement, and also it needs to be a specific PhD student that is named in the application. So here deadline is 7th of April for the industrial PhD student positions. So if you don't already have some contacts, it will be difficult to actually make up the project and apply for it, but maybe not impossible. But you need both the industrial partner, you need to define the project, and you also need to have the academic partner working together on the application. So thank you for your attention, and maybe there are some questions.
Yeah, thank you, Esben. Super interesting talk. Like, you covered so many different angles. Super interesting to hear about your personal journey, and then about your work in AstraZeneca, and about these opportunities that you presented us both in AstraZeneca and WASP. So I hope participants are taking notes, because every speaker has some different opportunities to present. So very informative event. And yes, I see a lot of questions popping in from the audience. So Antonia asks, can the algorithm take into account the limitation of chemistry possible to synthesize the chemical compounds?
Yeah, it's actually an interesting aspect of the algorithms. As I said before, when you train on a data set, you get molecules that are similar to that data set. So if you train it on molecules that is easy to synthesize according to various scores, you usually also generate compounds that are easy to synthesize according to the same kind of scores. Whether that holds in practice is a different aspect. We have seen that some of these molecules that we suggest in design are a little bit trickier for the medicinal chemist to make than maybe what they usually do, because it's very unbiased when it designs the molecules. It doesn't think, I have to synthesize this, right? So sometimes it knows the rules of chemistry, how to combine the atoms, and then it combines them in a little bit different way than the chemist would like it to actually make the compound. And then sometimes we end up with what is called AI-inspired compounds. Oh, we have to change it a little bit so that we can actually make it practical. But it's also an aspect we're working very actively in, trying to build in synthetic knowledge into the AI algorithm. And we have done that in a couple of different ways. One is a retrosynthetic planning algorithm that takes these compounds and then feed back into the reinforcement learning. Okay, I could make that. This seems difficult. Okay, this seems, and that can help. Other types of proxy scores trying to predict if it's easy or difficult to synthesize can also help in that aspect.
Thank you. Then another participant asks, how accurate is the novel compound generation and drug discovery? And what are some of the limitations with predictive chemical tasks?
Yeah, I'm not sure what you mean by accurate here. We make the compounds that fulfill the desirability score. And the problem is, of course, how good are your scores when you do that? And I think especially in the early parts of drug discovery, you may not have so much data, for example, building the project-specific activity model. So there you have to make do what you have, right? You can maybe build, if you have a couple of known actives, you can build a QSAR model around it, a quantitative structure-activity relationship model, and use that. Then you design some compounds, we go and synthesize and we test them, and then we start to learn a little bit more about the structure-activity relationships around it. So it really depends on the accuracy of the model, but as the project progresses through iterations in the usual DMSA cycle, we see that we learn more and more. So we become more and more accurate in the predictions that we make. And what the whole thing was about was to limit the number of cycles we have to do in the so to speak, normal design, make, test, analyze cycle. And we can see that the novel design algorithms, they actually successfully do that. We lower the number of cycles, but then sometimes at the expenditure, we need to work a little bit more on the chemistry side.
Thank you. Thanks a lot. Super interesting to learn from your expertise. And we have some other questions, but unfortunately, we're running out of time. But thank you again, Esben, for presenting us this evening.
I can answer some of the questions in the chat.
In the chat? Okay, would be super great. Thank you. Yeah, and I'm happy to present our next speaker. So Nikolay Oskolkov. Nikolay is a senior bioinformatician at SciLifeLab and Biology Department of Lund University. He received his PhD in theoretical physics in 2017, which I find super interesting. And he switched to life sciences and bioinformatics in 2012. He has been working on projects varying from biomedical data analysis and single cell to evolutionary biology and hnDNA. His range of interest includes mathematical statistics, machine learning, and data science. So your background is super interesting, Nikolay, and we're very grateful to have you here tonight. And I will allow you to do your screen share now. I think you should be able to do that.
Thank you very much. Thank you very much, Enrique. Hi, everyone. Very nice to meet all of you virtually, and thank you very much for joining. I'm a bioinformatician from SciLifeLab and also Lund University in Sweden. And in my work, broadly speaking, I'm using computational methods for life science projects. And here I would like to share a few ideas, maybe give you some flavor of how exactly and where exactly in life sciences we can use complex models such as neural networks and deep learning. So I usually try to sort of find a balance in my work between simple models and too complex models such as neural networks. I try to follow the Occam's razor principle philosophy, which basically tells us that providing that we have two models with comparable accuracy, the one that makes fewer assumptions or simpler model should be prioritized. Yes, so this is a sort of basically means that well, neural network maybe should not be used as a first choice, and indeed in many real-world life science projects, simpler models they provide you good enough outcome, and I just encourage everyone to think about this balance and the Occam's razor principle. And so that's why for me, neural network is a tool in a toolbox. So it definitely has advantages. It's a very good technique to have in your toolbox, but it's not the only tool, and there are other interesting tools that maybe it makes a lot of sense to use them for other projects. So I usually try to sort of think about amount of data. When I select a tool from this toolbox, I think about amount of data and data type that I'm working with. And if I have too little data, maybe I will go for Bayesian statistics, the way they do it in evolutionary biology. If I have a decent amount of data, I can use traditional frequency statistics. And only if I am sure that I have enough data, plenty of data, then I still can use Bayesian and frequency statistics, but I can do much better than this with the machine and deep learning. So you need a lot of data. And the question is whether we have really a lot of data in life sciences. That's a problem. There is a temptation, or sometimes people tend to think about amounts of data, big data by disk space the data occupies, and then there is a temptation to say that maybe genomics is maybe one of the heaviest data types in life sciences. However, I think a little bit differently about large amounts of data. So I typically think about data in terms of multidimensional arrays or matrices. And for me, genomics or genetic variation data looks like this. It has two dimensions, N dimension of statistical observation, and P dimension of features. In this particular case, it's genetic variance. For comparison, you can look at the data metrics for single-cell RNA-seq data, gene expression data, and sometimes the amount of elements in those two matrices can be comparable. However, you see that those two matrices are dramatically different. And unfortunately, in sense of mathematical statistics, you actually would like to increase this n dimension and decrease p dimension. So if you have a great p dimension, like for the case of genetic variation data, this is what is called high dimensional data. And those types of data are notoriously difficult to robustly analyze with statistics. So very challenging. To work with high dimensional data is very challenging. That's why, for me, this single-cell RNA-seq data looks more like a big data. This is what I can call a lot of data when you have a huge n and small p. And here I would maybe say that this is not a big data. And here in this presentation, I briefly give you some flavor, a few directions where I have personal experience and where I think so those directions in life sciences are very promising, and sometimes it's pretty straightforward to use complex models such as neural networks. And briefly, those are microscopy imaging. I think everyone knows that automated microscopy delivers millions of images, basically. And we have convolutional neural networks that can utilize this high similarity between neighboring pixels on an image, and in this sense, dramatically reduce dimensionality of the data. Of course, neural networks are superior for image analysis, and this is not a discovery. But what is less known is maybe single-cell omics. It turns out that it becomes almost a routine to work with hundreds of thousands or even millions of cells. This is a paradise for machine learning. You really can go beyond linear approaches and use very, very complex models working with single cell data. Next, metagenomics is not a high dimensional data. Typically, you don't discover that many microbes per sample. At least known microbes. Shotgun metagenomics is relatively cheap, so many people can afford sequencing thousands of samples. So it's definitely a promising data type in life science for applying neural networks. And genomics can be also viewed as a big data, in my opinion, if you don't consider one genome or one individual as one statistical observation, but you simply slice your genome into pieces. So each subsequence, if you treat it as one statistical observation, you can imagine that you can have billions of these sequences. And this is a really big data. You can train a neural network and do very good, robust analysis on this amount of data. So now I briefly go through all these directions and give you some flavor without going, unfortunately, too much into details. But I hope you will still learn something useful. So a few examples. Microscopy imaging, as I mentioned, we have millions of images, and we can't look at all of them. We can't expect them visually. Fortunately, we have these algorithms, convolutional neural networks that can, without actually us looking at the images, they can locate and classify cells, whether they can give us coordinates of the cells. They can tell us whether it's a good, bad, or ugly cell. They can count cells. They can say something about cell morphology without actually looking at the images. So I think it's a very cool and useful and handy application that is already here now widely used in life sciences. Single cell, what you typically want to do in single cell analysis, you want to study heterogeneity of cells. You want to discover new cell types. That's why dimensionality reduction and clustering are two unsupervised, are two dominating techniques in this area. And often people try to visualize cells through this kind of plots, dimensionality reduction plots, where one point is one cell and different colors correspond to different cell types. So this is a blood cells, and this is principal component analysis. And you're supposed to see like 12 cell populations. You don't see them because it's a linear dimensionality reduction. So you can do it much better with, for example, neural networks, which can do a non-linear dimensionality reduction. For example, autoencoders can give you a better understanding about cell types you have in your data. So autoencoders, it's a special type of neural networks with this funny butterfly architecture, and it's widely used. It became super popular in single cell analysis because it's an unsupervised neural network, and single cell analysis is essentially unsupervised. Data integration. Neural networks are great at actually merging different heterogeneous types of data. Sometimes you have different types of biological information measured on the same biological cells. For example, methylation, genetic variation, gene expression from the same biological cells. You can try to use synergy of those different variables, different biological variables for better and more accurate description of biological cells. And there are technologies that actually can deliver multiple types of biological information from the same cells. And so this is what is called data integration problem. You have to somehow combine those different types of data. And neural networks, they naturally provide you a way to convert these heterogeneous data types into common non-parametric space where you can safely merge those different data types. And just one example is you can use maybe single cell RNA-seq and protein level information from the same cells. This is a technology called CyTOF. And by combining protein level information with gene expression, you might better resolve this purple cluster. It's a different cell type, which was not very visible if you were doing a single cell RNA-seq experiment alone. So this is a promise of and power of data integration. Metagenomics or microbiome analysis. There are, as I said, there are tons of metagenomic samples available, freely available. There is a Magnify project that you can download basically hundreds of thousands of shotgun metagenomic samples from there. There's a great resource, Human Microbiome Project. It turns out that if you sort of visualize samples based on microbial abundances in different samples, they tend to cluster by tissue they originate from. For example, these oral microbes and gut microbes. One point is one sample. So you can use, you can check what kind of microbes drive these clusters. You can find sort of microbe markers for each cluster, and you can take those microbial reference genomes, slice them, and use this data for training a neural network that can easily predict, without knowing where a sample comes from. It can, for any given sample, it can predict whether this is most likely oral sample or maybe gut sample or skin sample. Without doing alignment, without quantifying abundance, just very easily, if you have this trained neural network, which I kind of at some point did, and it was very successful in predicting where a sample came from which microbial community a sample came from, without actually knowing the truth. It did a good job. And finally, neural networks on genomics. As I said, you can maybe genomics is sort of not the best data type for neural networks, as I mentioned previously. But if you slice your genome into subsequences and use them for training, that's a big data. And here I just demonstrate one example from ancient genomics. Ancient DNA, if you're not familiar, it's sort of a very fragmented DNA, also damaged. There is a sort of increased transition mutational rate at the ends of those sequences. And one problem in ancient genomics is modern contamination, because actually the typical sample is a horrible mixture of modern and ancient sequences from different organisms. And here you can sort of utilize this power of neural networks to give you per sequence prediction of where does the sequence come from, not only what organism, but also whether it's an ancient organism or modern organism. And to do this, you have to use this idea that you have to put a label on each sequence. So if you providing that you have a good training data set, where you know that those are truly ancient sequences and those are truly modern sequences, you can train a classifier that also can be sort of, it's pretty straightforward to do it, and also look at why this classifier can predict the ancient status of each particular sequence. And turns out in my case that the signal was coming from the ends of the reads. So this is a so-called saliency map that gives you some information, how informative each particular nucleotide on the sequence was. And it turns out that the signal usually comes from the ends of the reads, and it makes sense because the deamination, this increased transition mutational frequency, it's typically located at the ends of the reads. So I think you got some flavor of how and where and in what directions in life science you can use these complex models, neural networks. So thank you very much for your attention.
Thank you very much, Nikolay, for a very interesting talk. Artificial intelligence sometimes can be like a buzzword, but it's really important to educate ourselves on where exactly it can be used and at what capacity it can be used in these days, and what challenges we have to advance it even more in the future. It's been really interesting for us. Like, you have a background in theoretical physics. So what made you to transition into life science field, if you could share it with us? It's a very interesting transition.
Yes, so basically a lot of open questions, a lot of interest in open questions in life sciences. I think that there are, it's sort of a topic for separate presentation, but briefly speaking, I was fascinated by these biological problems and amount of open questions. In physics, it's a little bit different. There is sort of well-established knowledge and maybe challenging to make breakthrough, while in biology and life sciences, there are a lot of unknown things where people have a vague understanding, and sort of bringing robust mathematics from natural sciences to life sciences. This is like one of my sort of goals. This is how I see my contribution to this area, that I can hopefully be helpful, providing my sort of physics background and mathematical training to hopefully contribute to life science projects.
Thank you. Thank you. Very interesting opinion that you have, and I fully agree that there are so many unanswered questions in life sciences, and maybe the problem is that biology is so not static. There are so many different factors where, even though we humans, we share 99.9% identical DNA, but we're all so different. And then epigenetics comes in, so which are also another mysterious type of field. But I believe that in the next coming decades, we will have a lot of answers, because a lot of different experts are coming into this field, such as you, from physics, theoretical physics into life sciences. Very inspiring. And so another question we have from Tim, and he asks, as you have mentioned Bayesian statistics, how advantageous is the use of Bayesian machine learning or artificial intelligence models to encounter sparsely represented feature spaces where n is less than p?
Yes, so I'm a big fan of Bayesian types of analysis. I think it's a very it's a very good approach when you have to do risky predictions. Sometimes in clinical diagnostics or predicting, I don't know, earthquakes, self-driving cars, when it's about death or life, you better be on a safe side and be less radical in your predictions. So traditional frequency statistics usually, you know, this p-value type of mentality, it gives you yes or no answer. So it is a black or white, while Bayesian never tells you whether it's black or white, but it is a black with some, you know, some confidence intervals. And then it's up to you to sort of decide whether it's a good enough evidence or not. So basically, I really encourage everyone to think Bayesian when it's about risky predictions, when the outcome of your analysis can be really important for people. You better not be mistaken. Yeah, I think I answered the Bayesian thing, and then there was about n greater than p or p greater than n. Sorry, I didn't get the last piece of the question. Could you please?
Yeah, of course I can repeat that. So Tim asks, how advantageous is the use of Bayesian machine learning or artificial intelligence models to encounter sparsely represented feature spaces where m is less than p?
Right. So exactly for high dimensional data. So I am afraid lots of or vast majority of data types in life sciences are high dimensional, to my experience, where but what Bayesian statistics gives you, it gives you priors. That's a way to compensate for the lack of data. So n smaller than p or p much greater than n means that you have a lack of data. You don't have enough data to do robust analysis. So you have to invent your data. The priors give you an instrument to kind of generate sort of a little bit of data, or sort of make your predictions less dramatic, less risky. So it's sort of an instrument, a very nice tool that is particular should be used for high dimensional data, simply because the mathematics becomes more robust when you by using these priors you can sort of generate additional data and compensate for the lack of data. Hope I answered the question.
Thank you. Then Hadi asks, can we use artificial intelligence to annotate single-cell RNA sequencing data?
Absolutely. I mean, the annotation, it's one of the sort of direct applications of neural networks. Sort of, if you have an atlas, so the mentality is the following: if you have an atlas of single cells with many millions of single cells, then you can train a classifier, not necessarily neural network, but any other classifier. You can train to sort of to learn these gene markers for each cluster, and then when you sequence another sample, you can quickly, by using this trained classifier without doing any dimensionality reduction clustering, you can quickly classify all cells in this new sample. You can quickly say, okay, it's mostly maybe T cells, or 50-50 B cells and T cells maybe. So you can quickly get some overview of what kind of cell populations are present in your unknown sample based on a trained model, a model trained on an atlas.
Thank you. Thank you for extensive and informative answer. And the final question, as we are running to the end of our event. So we have a question: can you trust artificial intelligence models for clinical diagnosis or patient treatment?
I think so. The thing is, what I learned in sort of working in biomedicine side by side with biomedical people is that they have really often, not always, but often they have a great databases. And so we are talking about a couple of % of Swedish population, maybe. So you have an information about interesting clinical variables from a couple of % of Swedish population, which is a great resource. And neural networks absolutely have a power to give good prediction, given this huge sample size. So I believe so. Sometimes I also see sort of naive ways when, you know, maybe there is a very unique cancer type, but there are only 200 samples, and people sort of asking, is it enough to do neural networks on them? This is an example of sort of naive attempt to use artificial intelligence, but often in biomedicine, in epidemiology, we are talking about hundreds of thousands of people. And there are these databases available for clinicians. And I believe this is where neural networks should be used even more. And people do work in this direction, as far as I know. There are many good collaborations.
Thank you. Thank you for your answers. And yeah, unfortunately, we don't have more time for questions. But thank you very much again, Nikolay, for informative presentation and great answers.
Thanks a lot.
Yeah, so thank you everyone for attending, and we will send you right now a survey link where everyone is welcome to fill in, and this survey will help both the Synapse and Hub AI to improve our future events, to create students even more great opportunities such as this one. So if you have any questions, or if you're interested to join both Synapse and Hub AI, we both have openings for new volunteers now. Do not hesitate to contact me for Synapse and Ravsan for Hub AI on LinkedIn. So thank you very much, everyone, again, and especially thanks to our great speakers. Thank you. Have a nice evening, everyone. Bye-bye.